Professor Eduardo Amorim, who will be joining the Department of Biology at California State University Northridge this coming summer. Currently, Dr. Amorim is a research assistant in the Department of Computational Biology at the University of Lausanne in Switzerland. So he is coming to us from Europe right now. Previously, he was a postdoctoral scholar in the Department of Ecology and Evolutionary Biology at UCLA and the Department of Evolutionary Biology and Ecology at Stony Brook. He earned his PhD in genetics at the University of Rio Grande do Sul in Brazil. And he studies some really fascinating topics, including the paleogenomics of the first Americans, population genetics of human disease, shared polymorphisms between humans and Neanderthals, and gene flow across continents. And he'll be here today talking with us about paleogenomic insights into human history and adaptation. So everybody, please join me in welcoming soon-to-be Professor Amorim.
Thank you very much. Thank you for the awesome introduction. It's really an honor to be talking to you and with the ASU Center for Evolutionary Medicine. I know a lot of people from ASU, and I really like the work that is being done there. So for me, it's an honor to be to have this opportunity to talk to you. So today, I'm going to talk about part of my work that focuses on using ancient DNA or paleogenomics to learn about human history and adaptation. As Katie just said, I'm a postdoc at the University of Lausanne, and my first slide here is a picture of the campus, just to brag a little bit. It's a very beautiful campus. We have here near us the campus per se, and then we have the Lake Geneva here in the back, and then all the way to the back, we see the French Alps. And that's not the view of my office, but it's similar. So it's really beautiful campus. Unfortunately, we are not going that much to campus, but anyway. So in the University of Lausanne, I focus on my research with paleogenomics, and specifically I work with history reconstruction and natural selection. Here we only work with humans and human pathogens. And one of the questions that we are asking is, for example, how humans adapted to high altitude. So, so in August, I'm joining the Department of Biology at Cal State Northridge. And for those who don't know, Northridge is here, the San Fernando Valley in Los Angeles. And there I'm going to be doing part of my research that's focused on DNA, but mostly the computation part, and my other research with modern day populations of humans, dogs, and wolves. So before going further with my talk, I want to talk a little bit about ancient DNA, just to make some things clear, because the title I used is paleogenomics. I guess, I don't know if everyone's familiar with, but I guess everyone is familiar with genomics. And so paleogenomics, it's a subfield of genomics that use DNA that is obtained from archaeological samples. So instead of getting DNA from blood or saliva, for example, we get it from bones. And this is an actual bone that I studied for one of my projects. And this is me in one of the very few times into the wet lab. My work is mostly computational. And to study ancient DNA, as you see here, we have to wear a lot of different protections. I have many layers of protections in my hands, my face, and in my body. And this is because ancient DNA is a very, we have very, let's say, low quality DNA and DNA present in very low quantities. So sometimes what happens is that some of our material is contamination, and it's not the DNA from the individual that we think or we wanted to get DNA from. But if it's so difficult, why do we study or why do we use ancient DNA for anything? The interesting thing about ancient DNA is that we can recover genetic variation that has been lost over time. What I mean is that, for example, you might have some sort of genetic drift that makes a variant be lost, or you may actually have like an extinction of a whole population, and you don't have any individual from that species or that population present in today, so you can get DNA from archaeological material. And ancient DNA has revolutionized how we study not only human history and evolution, but like mainly human, the evolution of human and history of humans. And I brought here 3 examples. I'm not an author of any of those papers. I just brought just like very famous papers in ancient DNA. One, there was actually 2 papers in this issue that describe the peopling of different parts of the world by among humans. The second one is describing the genome of the Neanderthal. The Neanderthal is a human species that is extinct now as a species. We have some Neanderthal genes that were introgressed into humans, and that's because different species of humans interbred. And also this idea that different species of humans interbred was also one insight that we gained from studying ancient DNA, which is represented here by this last paper. So today I decided to talk about 2 different projects, and I've been working on 3 different projects in ancient DNA, but mostly focusing on those 2. One is about more generally the Middle Ages, and I have a paper published on that came out about 2 years ago, and it's focused on the barbarian migrations. And the other part of my research is focused on South America. That's something I've been studying since my PhD in Brazil, and now I'm studying the similar questions, let's say, but using ancient DNA. And the questions that I tried to answer in those 2 different projects are very diverse. And I think that's very interesting to show how many different types of questions we can answer with ancient DNA. With both of them, I do try to learn about the demographic history of the population. So what are the sources of gene flow that are that form that population, for example. But I also look at more detailed things such as social organization, which is something very rare in ancient DNA studies, and it's becoming more and more common right now. And for the Americas, we are trying to look into adaptation. We have so many challenges to overcome, but this is also something that you could see with ancient DNA. And we are also trying to recover ancient pathogens to understand what ancient diseases those peoples had. So the first part of the talk, I'm going to talk about this paper that some people read in a journal club, I believe on Tuesday. And this is the paper that I published about 2 years ago. And just a quick background about what type of population and what period we were studying this paper. We were studying the so-called barbarians, and the barbarians were everyone. So they were living there in the beginning of the Middle Ages, more or less between the 6th and the 8th century. And barbarians were called barbarians if you were not part of the Roman Empire. And to be part of the Roman Empire was something very, there were many different rules that could make you Roman, but if you're not recognized as Roman, you would be recognized as barbarians. And the interesting thing about this period of our history, specifically in Europe, is that these peoples at this time, they didn't leave any written record. The Romans wrote a lot about them, but we don't have any texts that actually came from barbarian peoples. So everything we know either comes from a Roman source or from archaeology. So we thought that bringing ancient DNA to this, to study this period and this period of history would be interesting because we could learn new things about these peoples. And the third thing is that, as I said before, like barbarians were these people that were not Romans, but they also are described in the Roman texts as peoples who invaded the Roman Empire. And some scholars nowadays, they associate the fall of the Roman Empire in the West with the barbarian invasions. And one of these groups of barbarians is the Longobards. And one curious thing about the name Longobard, it means long beard. So that's not how the Longobards called themselves. It's how the the Romans would call those peoples. We don't know actually how they actually named themselves. So we don't know much about their history, basically. And this is just a map showing a bunch of different barbarian groups, Franks, Anglo-Saxons, Vandals, and so on. And most of them come from North Europe, at least according to Roman sources. And they, at certain point in history, they invade the Roman Empire, which is depicted here with this like pinkish salmon shade and the brown shade. So, oops. Okay, so because of this, because we don't have a lot of text about those peoples, there is a lot of different questions that are remaining open. One of them is if those people actually had some origin in the north of Europe or somewhere else. The other one is actually if they came from regions that are near the Roman Empire or from really distant places, like sometimes they are described as coming from Asia. And during this immigration from wherever they are to the Roman Empire, how did they interact with peoples that they encountered on the way? Were they only killing those people, or they were also integrating those people in their society? And who were those people that they integrate, and who were those people that they didn't absorb into the society? And the last one is how those militias or how those raid groups were formed. Were they formed by only males or by males and females? How are those people related between them, and so on? So today I'm going to focus on the last two questions, because those are the questions that relate to more internal aspects of these historical societies. And the first ones are more related to long distance migration. And those are questions that are usually approached in many other ancient DNA papers, whereas the questions that are more focused on internal dynamics of these populations are more rare. So because of that, I'm going to focus more on those two last ones. And for this paper, we studied two different archaeological sites, one in Hungary and the other one in Italy. Today, I'm going to talk mostly about this one in Hungary. It's called Szolad, and it's located in West Hungary, in a place where at this time, the end of the 6th century, this belonged to the Roman Empire. And it's a province called Pannonia, for those who are familiar with Roman history. So for this cemetery, we did something that we call dense sampling, which is also very rare in ancient DNA studies. Instead of just sequencing or choosing a couple of individuals for each archaeological site and covering many different archaeological sites, we decided to cover only 2 archaeological sites, but trying to get DNA from all individuals. That's what we call dense sampling or high resolution sampling. And we, by doing so, we had 45 graves. We tried to get DNA from all 45, but we only get for 37, enough DNA to do genomic studies. And we carbon dated about 5 individuals, and all of them dated to the 6th century. And from the 37 that we managed to get DNA from, 10 best ones, we submitted to whole genome sequencing, and we got 7 to 14x coverage, which is pretty high for ancient DNA studies. We can actually genotype, type genotypes instead of just like single alleles, which is more common. For the other ones, the 27, we didn't have very good DNA quality, so we got this, we applied this method called SNP capture or SNP enrichment, where we select specific regions of the genome to sequence. And in our study, we sequenced over 1 million SNPs. And together with this information, we used to write our paper a lot of different informations coming from other archaeological sources, such as material culture, some anthropological measures to detect injuries, to detect age, for example, diseases. And also we had stable isotopes that indicate where an individual was born, and also what type of diet they had. So today, most of the things I'm going to discuss focuses on their material culture. And here I have some photos of some elements that I'm going to be discussing later. One of them is this, on the left side, we see some sort of like brooches that were buried with this individual. We also see one individual in Sola that has this wristband. Just like some examples of like jewelries that these people were buried with. We also see some graves that had this structure around it, which is like a circular ditch. The grave is in the middle. And we have also some individuals that were buried with different types of weapons. We have swords, spears, knives, and shields. So I'm not sure how familiar is everyone with this type of genetic analysis that we do. So I put this question here: how can we use genetic data to reconstruct our history? And here I gave some examples of methods that we use. We either usually we try to find relationship between individuals and populations. One of the relationships could be family relationships, or it could be between individual and population, could be how, what are the different ancestry components of that individual. And that's pretty this is exactly what those like 23andMe and Ancestry DNA, they do with us. This is an actual ancestry, my ancestry composition. So I have like 76% of European component, and most of it comes from either Spain or Portugal. I'm from Brazil, so it's Portugal in this case. I have 13.6 sub-Saharan African, and so on. I have some ancestry from different other continents as well. And we can do the same thing, but with ancient individuals. So when you know nothing about that individual, we use like if she or he was buried with something, but now we have a more complete, we can now have a more complete picture of how this person, more information about this person. And that's what we do. And instead of getting this like very wide panel with many different populations, I focus my analysis on 3 populations of European descent. One is Northern Europe, the other one is Southern Europe, and the other one is Western Europe, which is the Iberia Peninsula. And one second. So one thing that a lot of people ask me is, okay, so I use modern European reference data sets to use as a proxy for these ancestral components. And some people complain, they say, oh, why do you use modern Europeans and not ancient Europeans? We did that because at this point in Europe, we could already see some difference between northern and southern individuals. We know that because of ancient DNA. So I use this fact to see what is the composition of the samples that I was studying. And what we see here is for each individual, before I had for me this circle, right? For here I have for each individual, I have a bar. And the bars that are 100% blue, it means that they are 100% Northern European. And we see 2 shades of blue, just to represent 2 different ancestry components from Northern Europe. One is from Finland and the other one is from other countries. It's more widespread. And in red, we see the individuals that are Southern European, like SZ19, for example. It's 100% Southern European. And then we see a bunch of individuals that are mixed, like half of the population is mixed, let's say. They have some sort of a blue component and the red component. And those that have the red component sometimes have the green. So here, my next question, so one thing that we can observe from this study is that those peoples were very diverse. They potentially incorporated different peoples in their society, right? Some people coming from Northern Europe, some people coming from Southern Europe. So the next step was to see how these different ancestries were distributed in the cemetery. So Yes, and how did the archaeology, the material culture was related to each ancestry as well. And what we see here is the map, and the same map that I showed before, which with different graves here with some numbers and some small drawings. And we can trace this almost perfect line here in the cemetery that divides individuals with Northern European ancestry and individuals with Southern European ancestry. There are two exceptions. Those are two genetically female individuals, and they were all buried with jewelry. Whereas people from the like below this line, they were not buried with any type of jewelry. So comparing those two groups, besides the different genetic ancestries, they also have different material culture. The ones in the north have deeper graves. They have structured graves, some sort of like with chambers or sometimes with ledge walls, for example. And they also have furnished graves. All individuals that are buried above this line, they're adults, they were buried with something, usually a weapon or some jewelry. And the ones in the south, they were not like that. Their graves were very simple. They were just a hole in the floor, and they didn't have any furnishing. They didn't have any type of jewelry or anything. So this is just a photo that I showed before to show the different material culture elements that we analyzed. And what it shows, it suggests to us is that not only those people were very diverse, they had at least two different populations inside that society, but they also had different material cultures. So we could think that they were either people with different status, like social status, or different roles in the society. Maybe the other ones, some were more like peasants, the other ones were more like warriors, and so on. So another way that we can look at genetic relationships is to look for biological kinship. So to know who is the grandfather, or the grandmother, the mother and the father of each individual. And to do that, we use this software called CEMinar that uses ancient DNA specifically to reconstruct family relationships. And what we can see is that we found 4 groups of people that were related, and they were always buried beside each other. This is very interesting for who studies archaeology, because they were always very interested to know, like, oh, who is related to who? Yeah, so there is some sort of information that you get from that. They were aware of this type of kinship as well, for example. And the curious thing is that one of the biggest families that we have is this one here in the northern part of the cemetery. They had 8 individuals. And here on the left, you can see how they relate to each other. We have 3 generations, starting with number 24 and then going all the way down. And for those who are not familiar with this type of plot, of heredogram, the squares are male and the circle are female individuals genetically. And what we see here, and the little bar on the left tells us what is the ancestry component of that individual, and it's mostly blue, indicating that they come from Northern European genetic background. And the interesting thing about this family, not only that they are the largest one, the only one with 3 generations, but they're also rich in the grave furnishing. They have types of objects that no other grave have. For example, one of them have a scale, for example, to weigh things. And this is thought to be the person who was responsible for doing commerce, for example, to selling stuff. That's what the archaeologists believe. And also one of these individuals had a horse buried with him. Was number 13. So, but the most curious thing about this family is that we didn't find any adult female that were buried there. And when we found that, we saw, wow, that's really interesting. So we didn't get DNA for all of the individuals, but we actually got at least mitochondrial DNA. So we could rule out some of the female individuals being the mothers of one of those individuals. And in fact, we didn't see where the mothers are buried. We we were speculating a lot about that. What does it mean? Does it mean that they were not buried there? They remained in North Europe, let's say, or were they killed and buried somewhere else, or I don't know, or they not killed? We don't know. But the curious thing is that 1 year after my paper was published, this group from Max Planck Institute in Vienna, they published this similar study with trying to establish kinship between individuals from different cemeteries. And they found exactly the same thing in 3 different cemeteries. And the oldest one is actually 3,500 years ago. So from 3,500 years ago until 1,500 years ago, we observe at least 4 cemeteries that have this type of structure where the mothers, the female related people, were not buried together with the men, and they were not buried at all in the same cemetery. We still don't know what it means, but it does show us that paleogenomics, ancient DNA, can shed new light into the behavior of historical human groups. So those things were not obvious before when we didn't have ancient DNA, and now we have additional information to understand how ancient or historical populations behaved. So this is one slide that I want to summarize. Let me just drink a little bit of water. This is just to summarize 3 points that I approached in this work. The first one is that the Longobards, these peoples that we studied, they are genetically very diverse. So we found at least the presence of at least 2 different groups that that are present in that population. And that indicates that whoever the group, the half of the group that came from North Europe, when they migrated southwards towards the Roman Empire, they seem to have incorporated all the peoples, local peoples maybe, along their way. And these different ancestry groups also correspond to different mortuary practices. We see some that were buried with weapons, for example, and some that were not. And this could potentially be reflecting the fact that those peoples, those 2 different populations, had different roles in the society. And we don't want to do any speculation here, but because with ancient DNA, we can't answer what roles they had, but it does seem that they had different roles. And we also find, the last point is that we find evidence for a central family, the central role of one specific family. And the funerary or mortuary practice for this family was very distant from the others from the same cemetery. Not only that, but this family also lacks adult females that are buried with them. And we don't know what this could be showing. I'm happy to discuss that in the end, if anyone is curious. But just to wrap up this first part of the talk, I'm still interested and still trying to work with ancient DNA from the Middle Ages. It's a period of history that there is a lot of archaeological sites that are available and haven't been studied from the genetic point of view. And I have access to one of those that it's called Priory Orchard, Godalming, and it's in the UK. And we found human remains for over 300 individuals. And all of them are dated to around the 11th century, which is in the middle of the Middle Ages. So one of my ideas is to build this time series data sets for the Middle Ages. So I have already something in the beginning of the Middle Ages. I have something in the middle I plan to have soon. And then to get more data in the end of the Middle Ages, for example, and maybe try to understand allele frequency trajectories across time and try to associate with selective pressures, for example, with outbreaks of the plague, for example. But only looking at Priory Orchard Godalming, we have a lot of interesting things to do as well. We don't know who are those people that were buried there. So everyone who lives now at the city, they were not aware that some other people lived in the same city with them before them. And this is also potentially a mass grave. Mass grave is a typical thing from the end of the Middle Ages when we had the Black Death. So a whole city would die and they would dump everyone in a big how to say, a big mass grave. So, and they would leave the city. So they didn't want to bury each one of those in a specific place. They just put everyone in a big grave. But this is common for plague, and this is in the 11th century. So and the plague was there in the 15th century. So we don't think it's a plague. So it could be another deadly outbreak, another deadly disease. Or could it be an early outbreak of plague? So anyway, it's very interesting that we could study that. This is just an example of the quality of the DNA that we find there. The blue bar represents endogenous human DNA, and the green is everything else like bacteria or any other environmental DNA that is there. And we find 3 to 16% of endogenous human ancient DNA there, which is very promising. And this finishes this first part, which is focusing on Middle Ages, specifically on the barbarian migrations. Now we can go to the second part, which is shorter than that. And it focuses more on the research that I'm doing right now. And when I was invited for this seminar, it was a while ago, I thought by March, I would have a lot of results to show. And unfortunately, I don't have, but I do have some things that are very intriguing and shows the potentiality, like shows that where we're investing for this research is really could give us very nice and really interesting work. So I will just do again a quick background. We have, and it's about the peopling of the Americas. So I'm talking about here South America, and here I'm going to talk more generally how the Americas were peopled. And this is a paper from 2008. It's pretty old, but I really like this figure. I always use in my talks because it explains very well, I think. So we see that the Americas, well, this part A here, the history, we see it's something about 40,000 years ago when there is an expansion of an East Central Asian population towards this region here, which is between North America and Asia. And we call this region Beringia. We know, some people know, have heard about the Bering Strait. So the Bering Strait is what is left from this subcontinent. And before, the level of the ocean was lower, so you have more land exposed in this region. So you actually could inhabit, you could live there. And those peoples, they migrated from East Asia. They occupied the Bering Strait or the Beringia for many, many years, for maybe 20,000 years. And they were a very small population. And because of that, because of their size and because of the amount of time that they remained there, we characterize that as a population bottleneck. So with this population bottleneck, this reduction in the population size in comparison to the ancestral in Asia, we start to see a lot of variants that are lost. And then when these peoples, they enter the continent and they expand into the whole continent, you start to see new variants that are unique to Native Americans. So because of this moment, we are able to differentiate what's Asian and what's who is Asian and who is Native American, for example. So about 18,000 years ago, we have the last glacial maximum. Correct me if I'm wrong, but it's 18,000 years ago. We have the coldest temperature, the last coldest temperature that our planet had. And then the temperatures start to rise and the ice sheets start to melt. This white thing here is an ice sheet. So people were there in Beringia and they couldn't enter North America because they saw a bunch of ice. They couldn't cross that. But when the temperature rises, this ice starts to melt and we see that humans start to people the new world. So we see a bunch of different archaeological sites dated to 14.5 thousand years ago that are present in all the Americas. We usually don't see, I might be wrong, but I don't think we have anything that is much older than that. So that suggests that the Americas were peopled around that time. So 15,000 years ago. And then this population spread all over the continent in very, very short time. So the oldest archaeological site we know in South America, I might be wrong, but correct me if I'm wrong, if someone knows, is in Chile, and it's 14.5 thousand years ago. So that shows that the peopling of the New World was very fast. Is someone? Ah. And yeah, so related to the peopling of the Americas, I studied this since my PhD, and I focused again on two different aspects. One is history reconstruction, the other one is natural selection. Here are two papers that one that I first authored, the other one I'm co-author, that are my last papers on each two subjects. So one, we studied the dispersal and colonization of the coast of Brazil by Native Americans. And the other one, we studied the signatures of natural selection in Native Americans that are related to diet. And my current project is not related to those two. It's actually something that is focused on ancient DNA. And here I'm working with Ana Sapo Malaspinas, who is my postdoc advisor. And we have this awesome collaborator, Lars Fahrenschmitz from UCSC, who is a specialist in Peruvian populations, archaeology, and genetics. So we are analyzing part of his collection. We are analyzing 70 samples from different regions in South America. Here we see all those black dots represent one collection site, one archaeological site. And these samples, they are all pre-contact. So they are about, the youngest ones are like 600 years old, and the oldest ones are about 8,000 years old. And the questions we want to ask is about the history of the ancient Moche, which is a Native American group that lived in Peru. We are also concerned with understanding how adaptation to high altitude took place. And why is it important? Because a lot of high altitude populations, they have this genetic adaptations to live in high altitude. And they, other people that are not adapted to those, we would have a lot of different health problems, including problems in pregnancy and other diseases. And the third thing is that we're screening for ancient pathogens. So a quick, I'm going to talk a little bit about each topic very quickly, but most of the results we have now, it's for the Moche. And we have 14 samples that are dating for at the 5th and 6th century. And some of the samples that we have, one of the samples that we have is from the Lady of Cao, or Senhora de Cao. And this individual was seemed to have been an important leader, like a queen of this society. And you can see here how rich is like her grave furnishing. She has all like this coat with several plates with metal. She has a huge crown. She has a nose ring. I don't know if you can see, but this is a nose ring. And she has all this, and a lot of things that are like extremely precious, and that shows that they had a really developed way to work with metals. And although they were very developed society, they didn't leave any written record, which is common in many populations from the world. So we don't really, we again thought it would be interesting to work to understand more about this population. Oops. Okay. And one thing that we do in ancient DNA is this type of PCA plot where we see how, based on the distances between different individuals in this plot, we can see how our the ancient individuals are similar to present day populations. And here I use present day populations from Ecuador, from Amazon, from the Andes, and North Peru. And not surprisingly, those individuals that are Moche, they are grouping with individuals from Northern Peru. I'm gonna go back here just to show where the Moche are. So this is North Peru, so we would expect them to group there. But the interesting thing is that I have 14 samples, some I didn't include here, but some we have two exceptions that are individuals that are too far away from this, so they don't look really like Native Americans. And to understand why they were behaving like that, I started to compare them with East Asians, Southeast Asians, and Pacific Island people. And what I see is that B10 and B5, two individuals that are here, they are actually between East Asians and Native Americans, which is pretty surprising, as I explained before, because the population was in Beringia for a very long time and it was a small population. We see this population bottleneck. So now we can differentiate what's Asian, who is Asian from who is Native American. So these two people, they have some Asian component in their genome, and it could be related to many different things. I'm gonna go there. But first, just showing that this person, I also did this type of admixture analysis to show that B10 has 48% of East Asian ancestry, and B5 has 13% of East Asian ancestry. And we came up right now, this is really early and preliminary results, but right now we have three possibilities for to explain that. And one is contamination, of course. And the other one is if it's a later burial. So it's this population is pre-contact, but maybe some individuals went there and buried some people with Asian ancestry or mixed people that are in a later time, like more recently. And the third thing is that it's an actual signal, and it actually shows some sort of gene flow coming from East Asia. And we tested the three, the two of them, the first two, we tested for contamination. Our data is not contaminated. It has some contamination, but it's very low. It's around 4%. It's pretty, it could be high for ancient DNA, yes, but it still doesn't explain why you would have 48% ancestry from East Asia. We checked for if it was a later burial as well. So we carbon dated these individuals. And we found that they are actually from the 7th century. So they are actually from the same population, from pre-contact populations. So we are left with this idea, which is that could be an actual signal. So it could be showing some sort of transcontinental, trans-Pacific gene flow, which sounds pretty strange, right? It's pretty surprising to if it's actually the case, but it's actually not something that it's something that has been described before, but not from East Asian ancestry. Those are two papers, one from 2014, the other one from 2015, that describe that. This one here, they showed that two Botocudo individuals from Brazil, they have 100%, so those are ancient individuals. They have 100% of the ancestry tracing back to Polynesia. So they could be either recent immigrants or children of recent immigrants from somewhere in Polynesia. And this other paper using modern data, what they show is that the populations from Amazon, like Suruí and Caritiana from Brazil, they share ancestry with other, with Oceanian population that is not seen in Mesoamericans or North Africans. We see that the dark reds indicates that they are similar. And we don't see these dark reds anywhere else. So these authors conclude, and similar to the other ones, that has been some sort of gene flow or migration through the Pacific. So we are still not sure what's going on, but what we are doing is sequencing those individuals again, because maybe there was something related, I don't know, some issue in the sequencing. So we are double checking if the sequencing will give the same results now, but it doesn't seem to be any sort of contamination. So we are pretty excited with that. And we also can see some other findings with this study. We found that the Senhora de Cal, she was related to two other individuals in the same place that she was buried. And in the second tomb, we don't see any people that are related to each other, but we do see some interesting pathogens. One individual has this hepatitis B virus, and the other all seven individuals from this tomb, they have this human parvovirus B19, which is very mild disease. It doesn't it doesn't have a really it wouldn't kill you, so it's okay to see that this virus is actually spread in the whole population. And yeah, so this finishes the first part, which is history reconstruction. I think we are approaching the end, but this is the last slide. I'm just going to say what we are thinking about doing with natural selection. And what we're doing is that we're building these time series data sets for different regions in the Andes. And this is one of them, North Highland. We have 19 individuals. And here on the y-axis, we have the time. So the 9 individuals here, 9 circles in the bottom, they represent 9 or 10 individuals that were collected in around 600 years ago. And then we have some individuals all the way back to 8,000 years ago. And we're going to merge this data set with present-day individual, present-day populations, data from present-day populations. And we want to infer natural selection. Specifically, we want to identify loci that are related to adaptation to high altitude, and try to estimate the selection coefficient and the onset of selection. And we are testing a few methods that are a couple of methods, actually 2 methods that allows us to infer natural selection using time series data. And we are doing some simulations to simulate our data, the type of data that we have, to see if we would have power to detect natural selection. And the idea is that if we want to learn what is the trajectory of the allele frequencies throughout time, and if the allele frequencies change a lot and very fast, this could indicate that this loci is under strong selection, and which is the red curve. And on the other hand, if the allele frequencies don't change a lot, we would conclude that it's weak selection. In this space, I finished the presentation. I would be happy to get questions from the audience. I would like to thank everyone who came to the seminar today. And I would also like to thank Katie, Noah, and Jennifer for the invitation and helping organize the webinar. Oh, yeah. Mm-hmm. Thank you.
All right. Thank you so much for that, Eduardo. Questions from the audience? And Stone.
Hello. Wonderful talk,
Hi.
Eduardo.
Thank you. Nice to see you.
Good to see you. So I have a question about the coverage of particularly the 2 individuals that were kind of wonky on the PCA, because a lot of times low coverage will result in that. And also, when you're looking at pathogens, which is totally cool, I love it. Are these teeth? Are these, what bone samples are being worked on there?
Yeah, so those are for the Native American samples, we have teeth. For the barbarian one, we had petrous bone. Oh, yeah, so we have teeth, only teeth.
Did you find any pathogens in the petrous?
I didn't look at that actually. Yeah. So this is something that we're doing here. We have Ana Sapho-Malaspinas has in many of the different works she collected a lot of data for humans, but now she's starting to look at the pathogens as well. Cool. And we are finding some interesting things potentially, but we don't have a lot of experience with that, so we are struggling a little bit. But about the coverage, we have very low coverage. We have, I'm trying to find here, but I know it's below 1x.
Okay.
Yeah, so I can tell you exactly how, but it's really low coverage. And but we do find at least 200, those 2 individuals, I have to check here, but they have at least 200,000 SNPs that we are using in comparison to the 1,000 genomes. So we think maybe 1x is not good, but we do think the number of SNPs it's a good number, and it's enough for us to infer this continental structure of the data.
It'll be interesting to see if when you sequence more, if the signal holds, because, and I just say this because Maria, in her work with the Puerto Rican samples, had also some wonkiness, and additional sequencing kind of did away with it.
And do you think was because the first sequencing was contaminated or something like this, or because it was low coverage?
It was low coverage.
Low coverage.
And there was one main one that just remained low coverage and stayed wonky, and we just decided it was low coverage.
Yeah, we don't have any. Everyone who says there is any type of trans-Pacific gene flow, it's always from Polynesia or Australasia. No one says it's from East Asia. So we want to be very careful. So I think, and we have resources to do that. So I think we will try to sequence that in much deeper coverage. I hope the signal holds.
Cool. Interesting. Thanks.
Thank you.
All right. Any other questions from the audience? Noah.
That was really fantastic, Eduardo. Thank you.
Thank you.
I had a question about the work that you're undergoing right now, that you're planning for the future, about high altitude adaptation through time, and sort of when these populations started inhabiting high altitude, and presumably that's when certain alleles started to undergo selection, strong or weak. It was a lot of the work you're talking about has to do with single nucleotide polymorphisms. Are you also going to be looking at other types of other signatures? Other genetic adaptations like structural variation and things like that? That would be putative targets of natural selection for high altitude adaptation.
Yeah, no. So that's very interesting. I wish we could, but we have so low quality data. I mean, it's not much worse than many other works in ancient DNA, but with ancient DNA, it's very difficult sometimes to reconstruct haplotypes. For example, we usually have only one allele being sequenced. We don't have a genotype. So there's a lot of things that are very complicated to do. We are hoping to do everything based on single nucleotide variants. So we have actually whole genome sequences. While we are sequencing everything that we find, we're not targeting SNPs only. We are sequencing everything. So we are hoping to see these variants, and we are using these methods that are based on allele frequency changes because of the limitation of the data. We have some samples that are like 4x or 6x coverage, but still not very good to infer genotype. So yeah, no structure variation and possibly no genotype. So we will be kind of limited.
Yeah, I wonder, do we know if there are any structural variations associated with high altitude populations?
So I don't. To study high altitude adaptation is it's a new thing for me. I actually don't know all the literature, but the most famous cases is Emilia Huerta Sanchez. She published a paper, and I believe other people also published in Tibet from populations in the Himalayas. And that signal is EPA's gene. I forgot the number, EPA's 1, I think. 1, yeah, and that's not a structural variation, if I can remember. And the interesting thing is that whenever you do this research with Himalayan populations, you do find this signal is very strong. But when you analyze other populations, like in South America in the Andes, you don't see signals that are replicated from one study to the other. So yeah, so there is still a little. It's more, how to say, it's more unclear what are the targets in South America as opposed to the Himalayas. And you know about Ethiopia, I guess I don't know. Maybe in the monkeys you study, they have structural variation also associated with.
Yeah, I mean, that's one thing we're looking into that and haven't really too strongly. But the good thing about sort of like low coverage sequencing data is that if you can identify, sorry, we're getting super into the weeds here, but if you can identify like a structural variant, you just need cover, like rather than calling it a genotyping an individual SNP, if it's a large enough region of the genome that's either duplicated or deleted, you just need to have sufficient coverage in that whole stretch of the genome, which means lower coverage sequencing actually will help. It's possible to pick those things up maybe a little bit more easily.
But the reads are very small. They are, in average, in my case, they are 45 base pairs. So we, I think there's like, I know some people that study structural variation with ancient DNA. Well, I saw a couple of papers about it, but I actually never worked with that, and I don't know personally anyone who has. So I think there are some issues because of also the size of the reads. So if you don't see something there, maybe it's just because there was no DNA covering that, or not because there was a deletion. Could be something like this.
All right, there's a final question that we have time for in this part of the talk in the chat. It's from Jonathan Cartsonis, non-anthropologist question. Can you look at subtypes of Hep B and use that information to understand population structure?
Yeah, so it's also it was a bit risky to present this type of this research because it's still so early and I have more questions and more doubts than actually answers. From the first part, I know much more, but the second part, I'm still very, I still don't, there's a lot of questions that we are, that we want to answer. We are at the stage with the viruses that we identify potential viruses in those samples. And but I actually haven't done any analysis with the viruses per se. So I actually also don't know what's the subtype that we have, unfortunately. But we will soon probably know. But I think you can use pathogens to learn how populations migrated as well. So if you have, for example, a new pathogen that was never described in that population, maybe you start to see at a certain point this pathogen being introduced. So it was possibly introduced by someone from outside that population, for example. So it's possible to do this type of analysis, and Stone has super interesting work on that. Yeah, I think I don't know much more about it, but yeah.
Wonderful, thank you so much. That brings us to the end of our hour. Thank you so much for presenting and making the time to virtually join us from Europe.